Hello and welcome to BIO300, Biochemistry. I'm Dr. Gor Sarkisian, and today we're going to discuss some of the chemical foundations and begin talking about some of the physical foundations of biochemistry. A biological system, a cell, is composed of a handful of chemical elements, which include a carbon. Obviously, we are known as carbon-based organisms because the backbone of all of the biomolecules is made of carbon. So carbon, oxygen, nitrogen, hydrogen, phosphate, chlorine, sulfates. These are the main components of basically what makes life as we know it possible. We also have several ions that are essential for life, such as sodium, potassium, calcium. These are involved in cellular transduction, cell signaling transduction. They are involved in neuronal impulse transduction, creation of action potential in the neurons. And these ions are essential for keeping the balance within the cell intact. It's a physiological process that all living things undergo to keep the environment constant, which is known as homeostasis. So as we've mentioned already, carbon is at the backbone of biomolecules. And carbon is a very versatile element. It has 4 valence electrons, which means it has 4 electrons in its outer energy level, and it can make 4 bonds with 4 different elements. Some of the most common bonds that carbon makes include carbon-hydrogen bond, which is found in everything. Lipids, proteins, sugars, you name it. Carbon-oxygen, which is also found everywhere. Carbon can also make double bonds with oxygen. Carbon-nitrogen, carbon double bond nitrogen, and also obviously carbon-carbon bonds, which could be either a single or double bond, and these are found again everywhere. And carbon-carbon bonds are found usually, well, are always found within the main chain of a molecule, and are also usually found in the side chains of a molecule. So due to the nature of a carbon atom to make 4 bonds in 3-dimensional space, carbon bonds can form what's known a tetrahedral arrangement. It's basically if, for example, we have a carbon-hydrogen bonds only, the hydrogen atoms would arrange in such a way that they are the furthest from each other. And in a 3D environment, the 4 bonds spaced at 109.5 degrees from each other, more or less, are the angles that create the most favorable distance between similar charged particles, such as hydrogens in this case. And that is depicted in picture A. Now, carbon-carbon bonds are interesting in that they have a degree of freedom, rotational freedom, with respect to each other. Now, there are other factors that may hinder such freedom, but in general, the bond itself has freedom to rotate. In this case, we have ethane, which is CH3 bound to CH3 through a carbon-carbon bond. And these two, even though they have a energetically favorable conformation, they are free to rotate. I must note that a single bond connection between carbon and carbon has an average length of 0.154 nanometers. Now in image C, we have a schematic 3D representation of a double bond. Okay? Now these bonds are shorter. They are about 0.134 nanometers in length, and double bonds do not allow rotational freedom. So a double bond forms a plane through which the arrangement of side chain elements, such as in this case depicted as A, B, X, and Y, they are situated in a single plane rather than in images A and B, in which they are actually in 2 dimensions. So we cannot talk about biochemistry without talking about functional groups. So what are functional groups? Basically, covalently linked carbon atoms in biomolecules can form along linear chains, branched chains, cyclic structures. And to these, basically the carbon skeleton structures, the so-called functional groups are attached. And these are common motifs through the biology, through the biological systems. And these are some of the essential functional groups, and I would expect you to know these. These are very important. And here we have a motif of carbon-hydrogen bonds, the methyl, ethyl, and phenyl groups. Phenols are cyclic. The methyl, ethyl are carbon. They differ in number of carbons in the group. Methyl, ethyl, very similar to that of methane, ethane, right? Then we have the nitrogen hydrogen groups, NH groups, which is the basic is amino group, right, and the derivatives thereof. And then we have the sulfhydryl groups and disulfates. It's basically sulfur containing groups, and in this case the disulfide is a very important bond in macromolecules, in proteins, because these bonds can be formed between adjacent amino acids, which are the building blocks of proteins. So amino acids can form disulfide bonds, whether these are disulfide bonds between 2 different constituents of a protein or between 2 different amino acids within the same protein. But these are very important in formation of the tertiary or the 4th level of structure in a protein. Basically, the protein folding and a 3D proper conformation of the protein. So here's a question. As I noted, the disulfide bonds are very important. Could you please tell me which amino acid is responsible for the formation of disulfide bonds? Other very common functional groups include carbonyl, the carboxyl groups, hydroxyls. So it's basically CH-CO, CO double bonds. ethers, esters, anhydrides. The other that you will come across very often, and it will become a very, very natural thing, would be the phosphoryl groups. Phosphoryl groups are essential to energy transfer in the cell, essential for making DNA, RNA. So these functional groups on this slide and on the previous slide are important to know because they form the macromolecules the way they are to function properly, to arrange themselves properly in a cell's 3D environment, and to interact with other molecules, thousands of other molecules, in a very predefined, very controlled way. Review question. Please describe situations in which the phosphoryl groups are used. Please list 3 specific examples. Now, in biological systems, the functional groups are found in many different molecules, and many different molecules can express a number of these different functional groups, and they're very distinct and very recognizable. Now, in this slide, we have a molecule called acetyl coenzyme A, or acetyl-CoA, which is a carrier of acetyl group, which is depicted on the lower right corner. Basically, carbon double bond oxygen, single bond CH3. And that is involved in many enzymatic activities. Its main function is to deliver the acetyl group to the citric acid cycle, the Krebs cycle, to be oxidized for energy production. Now, we will talk about the Krebs cycle later on in the course. There are many different molecules and many different chemical processes that are involved in the Krebs cycle, and Krebs cycle is essential for energy production. But first, we will learn the individual constituents of the molecules involved in these cycles that produce energy. So the take-home messages: we have a large molecule which is polyfunctional, contains several functional groups, and this particular one, the acetyl-CoA, is an acetyl group transporter to the Krebs cycle. So there are some sets of small molecules that are constantly present in living cells, in the cytosol of living cells. There can be hundreds of different small molecules, which may include amino acids, nucleotides, sugars, phosphorylated derivatives thereof. They can have tricarboxylic acids. We'll talk about these later. And most of these are polar or charged molecules. Therefore, they are in the cytosol freely floating. They're water soluble. They can be at quite high concentrations, such as up to micromolar. And given that these are charged, they cannot freely diffuse through the membrane in the cells. And there are certain proteins in the membranes between organelles, in the membranes of the cells themselves, that actively transport these molecules in and out of from one to another, to the other side of the plasma membrane. And these are very tightly controlled and are essential to the, let's say, the well-being of the cell. Now let's define what a macromolecule in a cell is. Basically, macromolecules include proteins, nucleic acids, polysaccharides. And macromolecules are basically polymers of smaller monomers of these molecules, and they are usually 500 Daltons or less. So in terms of proteins, it would be amino acids, nucleic acids would be nucleotides, polysaccharides would be simple sugars. So the number of polymerized units in these macromolecules could range from several tens to millions. There are some polysaccharides that just go on forever. The synthesis of these macromolecules is a major energy consuming activity of the cell. It requires a lot of work to put millions of subunits of individual components into a chain, into a polymer. Now, once a macromolecule is formed, it can be assembled with several different units of, let's say, in terms of proteins, several different proteins that can come together and complex to form the final product. For example, a ribosome. Ribosome is not only a single protein, it's an assembly. So briefly about the individual macromolecules. Let's begin with proteins, which are long polymer chains of amino acids. Some proteins may have catalytic activities to function as enzymes. Others can function as structural elements or single receptors, transporters, which can carry specific molecules in and out of cells. Signaling receptors could be molecules that respond to a chemical stimulus to trigger the cell to perform an action. So altogether, proteins are very versatile and probably are the most versatile of all biomolecules. The nucleic acids, such as DNA and RNA, are also polymers. They are polymers of individual nucleotides. The DNA stores the genetic information. RNA is the vehicle to drive the expression of the gene in the form of a protein. Now, that's not the only job that RNA does. Another important role of RNA, of certain RNAs, are catalytic in nature. In complex with supermolecules, they can catalyze certain biochemical reactions. Now, the polysaccharides are basically polymers of simple sugars such as glucose, and they have two major functions. First, obviously, is the energy. It's a storage for fuel, basically, for the cells. And the other, basically structural in nature. It could be as an extracellular structural element with a specific binding site for particular proteins, like the cellulose, for example, in the plants. And so it in certain organisms, short polymers of sugar, oligosaccharides, shorter sequences of sugar polymers, can attach to proteins or lipids on a cell surface, and they can serve as specific cell signaling molecules. And we will talk about this later because it's very important in certain pathologies also. So another class of large molecules are lipids. Lipids are greasy or oily hydrocarbon derivatives. They serve as structural components of membranes. They are energy-rich storages. They can be part of pigments, part of intracellular signaling. So as opposed to lipids, the individual monomeric subunits in proteins, nucleic acids, and polysaccharides are quite large. For example, for proteins it could be in the millions, for nucleic acids up in the billions, and in millions for polysaccharides. The individual lipid molecules are relatively small, up to 1.5 kilodaltons, and generally are not classified as macromolecules. However, lipids can aggregate together and form non-covalent bonds, and therefore form very large structures. One prominent such structure is the plasma membrane. It is, in the grand scheme of things, enormous in size. It's a non-covalent aggregate of basically lipid and protein molecules. Now, we've already talked about covalent bonds and functional groups, which are very important for biomolecules. But let's now look at a biomolecule in its 3D environment. So if one looks at the molecule as a 3D structure, it becomes apparent that the arrangements of the covalent bonds and functional groups that the molecule contains are also very important. By just writing out the chemical formula on a piece of paper, there is a large chunk of information that is lost. So basically, what is known as stereochemistry, it's the 3D structure of the molecule, also plays a very important role in biological systems. And as you may have guessed, with carbon-containing molecules with such a large degree of freedom that we discussed earlier, most of the compounds in the cell exist as stereoisomers, which, let's define again, are compounds or molecules with the same chemical formula, but different 3D configuration. As we will see later in the course, that the stereoisomers of the same molecule can have profoundly different effects, or many times one stereoisomer has a biologically active properties and the other one just doesn't. So let's dive deeper and talk about what exactly a stereoisomer is. So, but first let's talk about how to illustrate the stereochemical structures. Here we have three different ways to represent the same thing. The image A is the structural formula in perspective form. What does that really mean? Well, all the lines there from NH2 to double bond oxygen to OH are in a single plane, whereas the hydrogen is extending away from us, and the CH3 is actually protruding towards us, and those are the annotations of the stereo configuration of the element in this structural formula. The image B, which is known as ball and stick model, basically is informative in terms of the relative bond lengths and bond angles. And then we have the image C, which is a space-filled model, in which the atoms are shown in the form of the van der Waals radii. So basically showing what its energy distribution looks like. So the configurational isomers are chemicals with identical chemical composition, but different 3D bonds. Basically, they cannot be interconverted from one to another without breaking one or more covalent bonds. So what are the factors that result in configurational isomers? Well, these could be as simple as a double bond around which there is no freedom of rotation, or a chiral center around which the substituent groups are arranged in a specific sequence. So we will talk about the chiral centers soon. So here is an example of molecular conformation with double bonds. These two compounds here, the maleic acid and fumaric acid, are isomers. They have the same chemical formula, but as you can see from the image, they are arranged differently. And unless we break the covalent bonds between two carbons, essential carbons, we will not be able to go from one to another. And such breakage of the double bond obviously would require a lot of energy. So as such, that would be a chemical reaction, right? So which means these two are chemically different. So in case of the maleic acid, we have a cis conformation in which we have the two hydrogens on one side of the molecule. The fumaric acid is known as a trans isomer, in which there is a shift between, or the hydrogens are flipped from one side to another. Each of these compounds are well defined and distinct from one another. They each have their unique chemical properties. So the proteins that these compounds interact with are very different. A protein that can bind to maleic acid will not be able to interact with fumaric acid. Binding sites of proteins such as in enzymes. And this is the reason why the biological effects of malaric and fumaric acids are so distinct from one another. So as we noted on the previous slide, in order to transform from cis to trans conformation in a double bond isomers, one would need to input energy, and the molecule will go chemical transformation. Now, that happens quite often in biological systems. For example, in the event of light detection, which is basically coupled with absorption of light energy by 11-cis-retinol. The 11-cis-retinol, when it absorbs light at about 250 kilojoules per mole, is converted to all trans-retinol, which in turn binds to its receptor, which triggers electrical changes in our retinal cells, subsequently leading the nerve to nerve impulse and recognition of that event as perception of light in the brain. So now I think that double bonds are quite simple, right? Now let's talk about chiral centers. chiral centers are basically when four different substituents are bound to a tetrahedral carbon atom. If there are four different substituents, then they can be arranged in different ways. They can have two different conformations, therefore forming a chiral center. So this yields two stereoisomers with similar or identical chemical properties, but different in essential physical and biological properties. So the word chiral is derived from Greek word hand, because a carbon atom with four different substituents is asymmetric, similar but asymmetric, and it's you will see in the next couple of slides that it can be represented as a glove, in which a glove from left hand looks very similar to the glove from right hand, but you can't really put the glove from left hand to the right on the right hand and vice versa. So as we noted earlier, a molecule with one chiral center can have two different stereoisomers. Now let's generalize it further. A molecule with more than two, let's say an n chiral centers, will have 2 to the nth power of stereoisomers. Simple math. So here is a simple model of chirality. We have a molecule, a carbon, attached to four different substituents, A, B, X, and Y. And these can be arranged in two different ways. And these two different arrangements are non-superimposable. And therefore they're known as enantiomers. And the carbon in the center is known as the chiral atom, or the chiral center. So how does it work? Now, if there is symmetry, obviously, if we take the a mirror and reflect our molecule in that mirror, we will get a reflection that would be, if we rearrange the original molecule, would have been superimposable on that image in the mirror, as we will see in a subsequent slide. Now, if we take the original molecule and rotate it such that it should superimpose on a mirror, well, if it's a chiral molecule, it will not, as you can see here. We have the image indicated by a black arrow, the molecule, and the reflection in the mirror. They are not the same. They cannot be superimposed. So this is these two, the one in the mirror and the actual one, are actually an enantiomer isomers of each other. Now this is fairly self-explanatory. This molecule is symmetric, or otherwise known as achiral, because we have a carbon with three different substituents. So these cannot have two different conformations. However you put them, you will have a single molecule. So we have the original molecule, we have its reflection, and then if we take that molecule and rotate it to match that reflection, well, guess what? It will match the reflection. It is superimposable. And therefore, this molecule is achiral, it's symmetrical, it has no isomers, period. Now, from the last couple of slides, it was obvious that enantiomers are isomers that are a mirror image of each other. But besides that, we have other isomers, such as diastereomers, which are again isomers but not mirror images. On this image, you can clearly see the image 1 and 2 are enantiomers, mirror images of each other. The 3 and 4 are also enantiomers, mirror images of each other. And if you can see, all of these are the same molecule. But the molecule number 1 and 3 and 1 and 4 are not mirror images. They are diastereomers. Same thing applies for the molecule number 2, molecule number 3 and 4. These are also same molecules, but diastereomers, non-mirror images of each other. Now we have already stated that the isomer configuration of a molecule is very important to biochemical processes. So it is vital to accurately represent the structure of each biomolecule so that its stereochemistry is unambiguous. Basically, the structure is denoted precisely. Now, for compounds with more than one chiral center, the most useful nomenclature is RS system, basically right-left system. So in this system, each group attached to the chiral center or the chiral carbon is assigned a priority. And the priority is as follows here. From highest priority OCH2 to the lowest priority hydrogen. So to define the stereochemistry precisely, we have devised a universal mechanism in the RS system in which the chiral atom is viewed with the group of the lowest priority away from the viewer, if you imagine the molecule in 3D. So if it has a hydrogen, it's away from the viewer, from us. And if the priority of the other three groups, obviously there are four, right? So we put the lowest one away from us, we have the other three basically protruding towards us. If the priority of these three remaining groups decrease in clockwise order, then we say the configuration is R, which stands for rectus, the R for, or right for in Latin. If the order decreases in a counterclockwise order, the conformation is said to be in a S, which stands for Latin sinister or left. Please note that you need to know this very well. This is very important. It's going to come back at you over and over. And believe me, I will ask you questions about this. So to illustrate what I just said, here is an R and an S conformation of two isomers. As you can see, the 4 here is the lowest priority, okay? And it's pointing away from us. And then we have 1, 2, and 3. And on the left side, we have counterclockwise 1, 2, 3 arrangement. Therefore, it's in an R conformation. On the right side, we have 1, 2, 3 counterclockwise. Therefore, it's an S conformation. So here's an example. But besides the RS system, we have another naming system for stereoisomers known as the L system. The L is basically synonymous to S. But we will talk about the DL system later. This is to illustrate an example of S stereoisomer. But if we have a molecule that has a single chiral center, the S and L and D and R are interchangeable and they do not make any difference. Now, when the number of chiral centers differs, then we start seeing some differences between these two naming systems. But here, now we have a hydrogen in this molecule, right? As you can see on the left. Now let's put that hydrogen to point away from us. So it's going into the monitor. Then the next higher priority is OH, right? And then we have the second highest is the CHO. Okay, and then the third is CH2OH. So it rotates counterclockwise, right? And therefore it is an S conformation. And this applies to all of these kind of stereoisomers, and the logic is very solid, and I'm sure you'll be able to tell me if molecule is an S or an R conformation in a heartbeat. Now, as we have already noted, a single bond between carbon atoms allows a freedom of rotation. Now, it doesn't mean that it rotates freely. Now, it still has certain configuration that is of lower energy than others, such as the staggered configuration is of lowest energy, and eclipsed is of highest energy. So the molecule tends to be in the staggered form. So if the torsion angle is 0 to 120 between these two groups, right? Then it's the eclipsed form. And if we have the torsion angle of 60, 180, and so on, then we have the staggered configuration, and it is the more preferred configuration of this particular molecule, which is ethane. So, as we saw on a previous slide, the energy difference between the two different forms is very little. It's about 12 kilojoules per mole, which means that these molecules can basically interconvert at megahertz frequencies. Therefore, in a homogeneous solution, it is impossible to separate the two forms from each other because they're basically the same thing. And they go back and forth at incredibly high speeds. But when one or more of the hydrogen atoms, depicted in white here, is replaced by a functional group that is either very large or electrically charged, the freedom of rotation around the C-C bond is reduced or hindered. This, in turn, limits the number of stable conformations of any ethane derivative that arises from such substitutions. Here is a review question. Let's assume that the frequency of conformational change between staggered and eclipsed form in this molecule is 1 megahertz. Please tell me what is the time domain of this transition between staggered and eclipsed form. So all in all, the biochemical interactions within the cell are stereochemical. Reactions such as within enzymes, they will react with one stereoisomer but not the other. Hormones with specific receptors, they are also bound to interact with their own specific isomers. Antibodies are specific for a form of antigen that they recognize only that particular form. This is due to the three-dimensional structure of biomolecules, the way they're formed, and it's a combination of conformational and configurational properties that enable interaction only with a specific isomer. A review question. Please describe the difference between cis and trans isoforms. Now, the laboratory experiments are not always 100% in correlation with what happens in a living system. So here are two terms here, in vivo and in vitro. In vivo means in a living system, in vitro means outside of the living system. So in living system, the chiral molecules are usually present in one chiral form only. So the amino acids, for example, in proteins occur only in L isomers, occur only as L isomers. Glucose occurs only as a D isomer. In the laboratory, when a compound with chiral carbon is synthesized, the reaction usually produces all possible chiral forms, a mixture of D and L forms. The living cells produce only one chiral form of these molecules. And it's likely because, well, it is because the enzymes that are used in synthesizing these molecules are also chiral. And remember, in a lab, we can heat things up to perform a chemical reaction. Well, in a cell, we can't. So the cells utilize enzymes to lower the energy of activation for a reaction to occur, which then conforms the product to a particular chiral form. So the binding site in the proteins is basically stereospecific. It can complement one isoform, but not the other. Just like the analogy I brought earlier of left glove not fitting the right hand. Now let's put all of this in the context of biology. Here we have two molecules, the R carbon and S carbon. As you can see, they're very, very similar with only one chiral center here. But in terms of biology, they are very, very different. They trigger two very different chemoreceptors in our olfactory system. The R carbon has a specific smell, the fragrance of spearmint. And the S carbon, surprisingly, has that smell of caraway. I'm sure you are all familiar with it. If not, I'm sure you will have some at home ask to smell these two different plants. It might sound strange, but it definitely puts it in perspective of how chirality affects the biochemistry. Here is another very interesting example. On the left, we have the active compound of NutraSweet, which is an artificial sweetener. And of course, it tastes sweet. But the one on right side, which is a D-phenylalanine conformation versus a L-phenylalanine on the left. The D-phenylalanine, it tastes actually very, very bitter. So even though they're similar, they can interact with their own receptor only, in which the first one triggers the sensation of sweet and does not interact with the receptor that triggers the sensation of bitter, and vice versa for the compound on the right. So, in summary, the binding properties of carbon, the 4 bonds that carbon can make, can create a long and elaborate carbon skeleton with number of different functional groups, which give biomolecules their biological and chemical properties. So also, as we've noted, a collection of several hundreds of small molecules are found in living cells. And these are many of the byproducts of metabolic pathways. And these are very good indications of evolutionary traits that have basically perfected the art of metabolism. These compounds are stored, they are reused, they are used as signaling molecules and whatnot. The biological systems tend to not waste too much energy. Another point is the proteins and nucleic acids are linear polymers. They contain all the information needed to actually fold upon itself to form a three-dimensional structure, therefore forming a biologically active molecule. Here is a review question. Review question 1. What is the difference between diastereomer and enantiomer? Question number 2. Which group has a higher priority in RS system, the OH or CH3? So the biological configuration can be changed only by breaking a covalent bond. For a carbon atom with 4 different substituents, the substituents can be arranged in 2 different ways, generating stereoisomers with distinct properties. And only one of these stereoisomers, as we have discussed already, is biologically active. Now, biologically active in a particular system. The molecular conformation is the position of the atom in space that can be changed by rotating about single bond without breaking the covalent bond. Now, the interaction of biomolecules is stereospecific. They require complementary match between the 2 interacting molecules for the reaction to occur, whatever the reaction is, whether signal transduction or enzymatic activity. These 2 systems need to come together in a stereospecific manner. Excuse me. Review question 1. Please explain why an in vivo effects could be different from in vitro experimental results. Review question 2. Why is the trace element ferrum important, and what biochemical processes is it involved in? Review question 3. What is the general purpose of acetyl-CoA?